I'd like to introduce first Dr. Gerald Spaeth, who actually had already a foot at Pasteur when he did his PhD, as I heard, then went to the States in Steve Beverly's lab for a postdoc. Then he made a zwischenstopp in New York for a few years, a faculty position between coming back to Pasteur in 2005, where he's leading the Laboratory of Molecular Parasitology and Signaling Unit. I think it's fair to say that Gerald is a Leishmania expert. He had a leading role in diverse consortia dealing with Leishmania, so EU-sponsored consortium, the Leish drug, and now he's currently a coordinator of two such consortium, TransLeish, and the other name we'll have to look about it. And we are all looking forward to his talk, tools and challenges for the next decade in Leishmania research. Thank you.
I will go over there, or
yeah,
and I will advance the slides just on the computer, maybe.
yeah, right here.
Okay.
And you have your pointer.
Perfect. So no pressure. The expert is speaking, and I think first of all I would like to thank, of course, the organizers and Eric for the nice introduction. The very first challenge is actually on this slide, it's actually the title. I mean, I was given this title, I have to fill this title, and I don't know whether, I can, I would say, fulfill your expectations that I will talk about the tools and challenges for the next decade of leishmaniasis research. I will try my best, of course. And this is a personal vision. It is not really. So, first of all, of course, I would like to present the project that we're working on. We're working on the protozoan parasite Leishmania, which comes in two different developmental stages. You have a promestigode parasite that resides in the sandfly insect, which then this parasite is inoculated into the mammalian host during a blood meal, where it's phagocytosed by macrophages, which then the parasite develops into these intracellular amastigodes, a differentiation that is triggered by pH and temperature changes. So based on that simple life cycle, what we actually are set out to do is to apply phosphoproteomics approaches, apply genetics approaches on protein kinases, to actually understand the biology of this development and eventually identify targets for anti-leishmanial chemotherapy. And I wouldn't be anyone without these people. This is the current lab, and some of the data that I will present today are actually generated by Olivier Leclerc, and the second part of the talk is mainly by Pasteur. So, talking about current limitations in Leishmania, first thing, of course, is the three big topics of clinical development. And I think here we can all agree that in terms of chemotherapy, there's a lot to be done. In terms of prognostic, diagnostic, marker discovery, the same. Vaccination is non-existent. So in terms of chemotherapy, one of the key elements, I feel, and I told you it's a personal view, is that we have actually to gain insight into parasite-specific biology and physiology, because this gives us the biggest therapeutic window to actually have a drug eventually that will act only on the parasite and not on the host. In terms of prognostic and diagnostic markers, and here I'm referring to markers that actually allow you to predict whether the parasite that you want to treat is resistant or not against the drug that you want to use. Here we are faced with a different kind of complexity, because these parasites are extremely adaptable to different environments. They change constantly their genomic makeup, which translates into different phenotypes, drugs resistance, tropism, and so on. So this challenges, of course, everything in terms of biomarker discovery. Third, of course, I think one of the major challenges in vaccination is actually to understand how the parasite escapes and subverts the host immunity, and eventually design ways to rescue host immunity. And I'm not going to talk at all about vaccination, because this is the DNDi, so we cannot do everything. But I think one thing that I just want to mention here is people need to actually study asymptomatic infection, because that's the key eventually to understand how we actually should protect people that are symptomatic. So as I said, I'm not going to develop vaccination at all, but I would like to focus right now on chemotherapy and the parasite-specific biology. So In terms of drug development, of course, we're facing a major problem that our parasites, the Leishmania, is a eukaryote just like we are. So obviously this provides some limitations in terms of finding specific parasite-specific targets. However, if you look closely into parasite biology, there's many angles that you can attack. So you have various features of the structure of Leishmania that are absolutely specific for this class of parasites. Another thing that we find quite appealing is these parasites do not have transcription factors, promoters, enhancers. Everything is expressed all the time, no matter what stage. And we're actually working very much on that question. And we showed that it's most of the differential expression happens on post-translational levels, of course. But what I would like to develop as a challenge today is coming in now, which is actually what I call the hypothetical proteome. So if you go on the DNDi database and you just query hypothetical protein, what you come up with is actually more than 5,000 hits of a genome that carries about 8,200 open reading frames, which means 60% of Leishmania is unknown, is parasite specific, does not have any homology to anything out there in any kind of database. And I think that one major challenge is, of course, to understand what these proteins are doing. Now, one thing that is quite striking, and that is the first approach towards these hypothetical proteins, is if you look what kind of enzymatic classes are actually missing from the Leishmania genome, you can find caspases, classical caspases are missing, tyrosine kinases, nuclear pore proteins, all these things seem to be missed in the genome. However, there are many papers that actually identify the enzymatic activities and activities of all of these things, which means there must be some kind of an alternative biology in Leishmania that runs these enzymatic reactions without that we bioinformatically can find the corresponding enzymes. So this is the challenge. Annotation and functional characterization of the Leishmania hypothetical proteome. Now, by no means I want to pretend here that we're working on that. We do not work on this, but I just feel it's a very important question for the community and, of course, for drug development as well. And what I would like to do today is just to show you that we are actually developing and applying for our own research on kinases. We're applying proteomics approaches, structural, functional, activity-based proteomics, and all along with these approaches, we always identify a lot of hypothetical proteins. So in a sense, we already start to actually elucidate a little bit more this proteome. And I just want to give you a couple of tools, how we actually, what we apply as techniques. So first series of experiments that we published over the past 2-3 years was dealing with Leishmania phosphoproteomics. Now, in order to gain access to the Leishmania phosphoproteins, very easy, you use this kind of affinity column where you actually put in a total extract that is a mixture between phosphorylated and non-phosphorylated proteins. Those that are phosphorylated will be retained, non-phosphorylated will be washed out, and then you can elute this phosphofraction. You can analyze these phosphoproteins from promastigotes and amastigotes either in a gel-based fashion or by LC-MSMS. And of course, once we identify the proteins that are phosphorylated in Leishmania, you can express this in a nice pie graph. And as you can see here, there's 44% of all the proteins were identified in these analyses that are actually covering hypothetical proteins. And as any smart scientist out there that we need funding, we actually discarded to work on these proteins because you cannot get funding saying, I don't know what this is doing, and it's actually no analogy whatsoever. There's no hypothesis that is behind these proteins, so you cannot get funding. What we did, however, we found extremely parasite-specific phosphorylation sites in Leishmania heat shock proteins and chaperones. We could develop a hypothesis and we could obtain, of course, funding. So the challenge is, how do we get funding for hypothetical proteins, which will be, of course, non-hypothesis driven? Now, this kind of affinity purification here in this case was interesting because we can now annotate these hypothetical proteins as kinase substrates that are phosphorylated. Of course, you can apply this on many different post-translational modifications, acetylation, sulfatation, and so on, in order to kind of use these affinity approaches to already annotate the post-translational modification of these hypothetical proteins. Now, what we developed in the lab over the past few years, and this is a different approach, so it's not really linked to the first one, is activity-based proteomics. So we would, what we decided to do is to actually identify protein kinases in Leishmania using an in-gel kinase activity assay. Principle is very simple. You actually have a gel that you cast, and while you're casting the gel in the gel liquid, you add a recombinant substrate. This substrate will be embedded in the entire gel, will not migrate in electrical field. You use this substrate-loaded gel to separate your Leishmania extract. You then renaturate, this is an SDS page, you renaturate the proteins in your gel, and you apply a radiolabeled ATP. And so what happens is any kinase that refolds and that can use the radiolabeled phosphate to add it to the substrate that is embedded in the entire gel, this kinase will reveal itself by transferring at the spot where it's migrating the phosphate to the substrate. So in other terms, the kinases reveal themselves by their phosphotransferase activity towards the substrate you add in the gel. Now, this has never been applied on Leishmania, but of course it was working very well. Here we have 3 settings, no substrate. We have one setting with MBP and one with beta-casein as substrates. These are 2 generic kinase substrates. What we can see with this technology, of course, is that you see not only substrate-specific differences, so each single signal is one or more protein kinases that can phosphorylate these substrates in the gel. So we have stage-specific and substrate-specific signals. Unfortunately, of course, the resolution of a 1D gel is not very high to actually identify these proteins. So Olivier, the technician that developed this tool in the lab, went on to adapt this to a 2D fashion, so by including a first step of isoelectric focusing, and then the rest was done the same. And as a matter of fact, one single band can actually be exploded into many, many spots, and I just cut out some parts of the gel here. We went on then to determine the proteins that are behind these spots. All of these identifications were then used in order to see what kind of function is actually enriched in our data set, and we found that ATP binding was enriched, which of course makes sense because we of course look at kinases. But again, when we plotted out the proteins we identified, we had substantial number of proteins with the different substrates or without substrate, which are actually hypothetical proteins. And here of course the question arises, are these hypothetical proteins do they carry kinase activity? Could these be alternative protein kinases? And I will show you in a little while how we actually address this question. Another approach in order to query the Leishmania kinome is actually to use this kind of capture compound, where you have a generic kinase inhibitor, staurosporine, which has a sorting function, so biotin, so you can actually filter out things by a streptavidin affinity column. You have a UV crosslinker to stabilize any kind of protein that binds here. And what we do with this compound, we actually put our Leishmania extract together with the compound and crosslink. And in parallel, we do the same, but we add staurosporine free as a competition control. And at the end, we then compare whatever is specifically bound in this assay and specifically competed away in this assay are specific binders. So this method, which we also published, I think, this year, again, if we look what kind of proteins we obtain using this kind of functional enrichment analysis, it's very specific. We got these kind of functions in our data set, kinase activities and ATP binding. And again, when we now look at the pie plot, we have, of course, a substantial number of proteins that have this ATP binding capacity. But again, we got a lot of proteins that are hypothetical. And here the question again arises, are these things directly binding to staurosporine? Or maybe they are just association partners? Or are these things maybe alternative ATP binding proteins? And this is the last approach that we're using for getting insight into the kinome. So here we have an affinity column coated with ATP, where you apply the extract. You, of course, elute out any kind of ATP binding protein that is binding to these beads. This is working very efficiently. We have here promastigote, amastigote extracts that we separated over 4 different ATP agarose beads. So this is 4 different ways ATP is linked to these beads. You see that we get very different patterns, and we already know that there's many different proteins coming off from the different beads. Again, we actually identify many, many hypothetical proteins. So, what do we do with these hypothetical proteins? They could be either providing insight into an alternative biology, alternative ATP binding proteins, alternative protein kinases, or they could, of course, just be associated with proteins that we pull down. In order to address this question, we have now established a collaboration with the Seattle Structural Genome Center in Seattle, and this is actually a high-throughput structural proteomics center dedicated to structure hypothetical proteins. And so we submitted a list of more than 20 hypothetical proteins that we feel very strong about, and they currently, they clone the gene, they will produce these hypothetical proteins as recombinant proteins, send it back to us, and we will, of course, with a purified protein, then test on our assays, are these proteins binding ATP? Are they binding staurosporine? And if we find binding, that would be, of course, a proof that there is an alternative biology. So to wrap up the first part, I don't know the time if I'm running late already. So to wrap up this first part, the first challenge is this Leishmania hypothetical proteome. I have, of course, to come up with a strategy. How do we actually cope with this? I believe that systems level analyses are required to really gain insight into the gene function and protein activity of these proteins. There are different ways you can do this kind of systems level analysis. I showed you a couple of proteomics techniques that allow you to already elucidate expression, modification, and binding activity. Of course, these information can be actually used in order to prioritize some proteins as already drug target candidates. Then the next step would be to do some kind of a high throughput biochemistry approach where you clone and express, you reveal the structure, the function, the activity. Once you have a handle on the activity, of course, this can be exploited for target-based screening assays. And finally, of course, we need to do the genetics, so overexpress, knock out these hypothetical proteins, do the necessary phenotypic analysis in terms of virulence and survival. And of course, this is straightly linked to drug target validation because your target should be absolutely essential. So I'm calling for a Leishmania hypome project similar to the Leishmania genome project, which means this kind of systems analysis should be done on a global level. So this needs to be done by a consortium of many, many Leishmania laboratories that are dedicated to that. Unfortunately, that does not exist today. I will not coordinate something like that. I would be more than happy to be part of it. But I'm coordinating something that leads me now to the second part of the second challenge, actually, which is our, I would say, failure to actually discover biomarkers for explaining drug resistance phenotypes and so on. So here just a very brief life cycle again of Leishmania. You know that we have, of course, not only the insect vector and the human host, but there's a lot of different reservoir animals where the parasite can circulate. So while the parasite is evolutionarily perfectly well adapted to cope with the sandfly environment or the mammalian environment through these differentiations, the parasite also actually can respond to changes in this environment. And this is just a list of changes that the parasite might experience in these hosts. So for example, it has been shown that parasites can adapt to arsenic in drinking water of the patients that you want to treat, and they gain resistance. Of course, the stronger your immune system, the more you will select out virulent parasites. It's kind of a, red queen race kind of thing. Then, of course, the genetic background of the human plays a role. The drug treatment plays a role. All these things imprint themselves on the Leishmania genome because all these things are selective pressures to allow the best parasite to grow out. Now, what does this mean in terms of the epidemiology that has been done until today? What happens usually is that you actually have your field isolate of interest, which means they are drug sensitive, drug resistant. You want to understand what is the basis between these differences. You adapt these parasites into culture, you mass expand these things, then you do your genetic phenotypic analysis whatsoever, and then you have some correlations that you find between different isolates, and you have your epidemiological conclusion. Now, I believe that this is heavily compromised due to the fact that you actually have to adapt the parasite to an environment that the parasite never seen in his entire life cycle, which means there will be other selection procedures going on, and this is actually proven in a model that we used with Pascal, our engineer. So there we asked the question, how well do in vitro models actually reflect real parasites? How is culture affecting parasite expression and genome makeup? We exploited a system that is very widely used in the Leishmania field, and of course all the drug screeners, they use Leishmania donovani, the LD1S, because it is growing extremely well in culture. It is converting into axenic amastigotes. Now, we were lucky enough that we had actually the same identical parasite recovered from Hank Murray from Cornell University when I was in New York, which these parasites were exclusively maintained in hamsters, so in a very natural environment. And Pascal once decided that she wants to actually compare the axenic parasites with the real hamster-derived parasites. And we had quite some surprises because first we used the high-throughput genome sequencing. I don't want to go into detail of that. Just to tell you that this way of gene sequencing allows you to count how many genes you have for a given gene. It allows you to count how many chromosomes you have. And here are the data of the genome sequencing of splenic amastigotes compared to axenic amastigotes. And all of these bars reflect the number of reads that you obtain for the different chromosomes. And I think you can immediately see, without that I explain more, there are a lot of differences in various chromosomes. For example, we have definitely aneuploidies or chromosomal amplification for chromosome 1, 5, and 3, which are specific for the axenic amastigotes. Even worse, we have a definitely very homogeneous trisomy in chromosome 26, which is shown here. This is because you have so many more reads. And of course, the tetrasomy of chromosome 31 is a very well-established fact, which actually allows us to anchor what is a 4N, a 2N, a 4, 2, and a 3N genome. Now, if we use a similar approach, but now quantifying the transcript abundance in these two parasite isolates, of course, it's even worse. So first of all, chromosome 26 shows that because you have a trisomy in axenics, many of the transcripts coming off from chromosome 26 are, of course, more abundant because you have 3 chromosomes. This is reflected. And of course, all of these other brushes that you see here refer to transcriptomics differences in axenics, chromosome by chromosome in red, and in splenics, chromosome by chromosome in green. So 2,500 genes are differentially regulated. We found deletions, aneuploidies. It's kind of a scary scenario. This was long-term cultured versus hamster-derived. We tried to actually reproduce this result in a shorter time frame by taking hamster-derived amastigotes, so the good ones, putting them into culture and just see how they evolve over just 20 passages. This is nothing. We did this kind of analysis, but for right now, I just want to show you the phenotypic results. What we see over these different passages is that in terms of doubling time, early passage parasites in culture, they grow very slow. And they are selected during increasing passage number for faster and faster growth, which means something happens that you favor certain parasites. Of course, those that undergo most likely genetic changes. Now, if you look in terms of what happens with virulence of these parasites, infectivity, in a macrophage assay, you see that the higher the passage number, the worse they survive. And after passage 25, our cultured parasites lost irreversibly virulence and intracellular survival in peritoneal macrophages. Now THP1 may be different, I don't know. The same was shown in a hamster model where only the early passage promastigotes kill the hamster, the later passage they're just inert or not establishing any disease. So this brings me to one of the conclusions here. What happens, and at least what we see in our culture system, that you have a mutual exclusive correlation between parasites that gain virulence are very poorly doing in culture and vice versa. Those that are really very well adapted in culture are doing very poorly in terms of virulence. And the reason is that you have all kinds of purifying and positive selections that lead to transcriptional changes and genomic changes. This, of course, is something that we have to consider when this genomic flexibility, when we consider drug screening models like us and our colleagues. And of course, anything in terms of current and future drugs, the parasite will be able to eventually, through genomic variability, create resistance. So this is a big, big challenge. But the other challenge, and this is the real challenge I want to raise here, is biomarker discovery. So how can you actually discover biomarkers if you rely on cultured parasites that are genomically totally diverging due to the culture? So the challenge can be met most likely using what I call a systems epidemiology approach, which means don't rely on culture anymore. Take the parasites straight out of the lesion, of the sandfly, and sequence the genome, sequence the transcriptome, and do your correlative studies on parasites that are not adapted to culture. Now, this is a very challenging idea, and I think that the Pasteur Institute, I have to do a little bit of publicity because we're here, we are actually in a prime position to tackle this kind of systems epidemiology approach and create a consortium. So I'm since 2 years coordinating a network to make people collaborate inside the Leishmania laboratories of this Institut Pasteur network. What is really fantastic in this network is that we have Pasteur institutes with a public health mission which have access to field isolates, and they have all kinds of collections already. And of course we have other Pasteur institutes which work with lab strains which are not so relevant, but they have, of course, all kinds of high throughput capable technologies available. And we have to actually come together. So thanks to this network, we actually were building a project which we termed Lice Shield. And I just want to very briefly show you what this Lice Shield is. It's actually using a systems epidemiological approach by having access to sandflies, parasite isolates, animal reservoirs, and patient sample through some Pasteur institutes, which then will be processed in collaboration with other Pasteur institutes and, of course, outside partners for high throughput analysis. And we hope that we will find markers that can be translated into application with various, of course, partners, including DNDi. And that this is not just on the paper, but that this is really going on. We have currently pending an application for a pilot study where we have all these partners with all their collections and the different phenotypic characterizations that they did, people that are working then downstream in high throughput sequencing and bioinformatics in order to investigate intra and interspecies diversity, understand the phenotypic and genotypic variability, and finally, of course, correlate this variability to different differences in, for example, tropism or in drug susceptibility. And eventually we hope that this pilot study will be expanded to a Lice Shield consortium that might be sponsored by the Horizon 2020 project. I may take over the scientific coordination here, where we actually apply systems approaches on to study transmission, how different transmissions actually affect Leishmania phenotype and genotype, and how this actually boils down to differences in disease development. And I would like to end this talk with kind of a wrap-up. So the Leishmania Research 2020, what I believe there are actually 3 major components, which means we have to bring together systems-wide analysis with disease-relevant field isolates and apply computational biology. This is not bioinformatics anymore, it's computational biology, because these times, with the tools available, you do biology by looking just at sequences. And I think there is something extremely interesting that happens in this interface, because bringing these things together will allow you 2 things to happen. First, you will actually have a tool to generate new hypotheses, which will, of course, incite fundamental research. But on the other hand, this will allow you to discover new biomarkers, which of course will feed into translational research. So I would like just, oh yes, and of course all of this can be brought together by creation of multinational, multidisciplinary consortia with private partners, non-profit partners, and academic partners. And I would like to thank the people that I'm working with, mostly people in the network and the various funding agencies. Thank you.
So thank you, Gerald. You even set a record already because regarding time, because you are perfectly on time. So
German.
This is great. I think your talk raised really well the challenging issue we are facing with drug discovery for Leishmania. I just noticed it. So in vitro, in vivo correlation, the biomarker issue, and you even touched to the partnership issue within the same organization, which I found quite interesting. Anyway, any question in the audience, please? We have time for 2-3 questions. Yeah.
I'm Ronald Gormos from Psychopolitics. I don't need a mic, I think I'm okay. I was wondering, I'm not very familiar with the subject, but is there anything known about the vectors? So my question is there anything known about the vectors that keep the parasite in the virulent state? And if so, could those factors be added to the culture to keep the parasite in this virulent state? And the last question related to that, could these factors, if they are known, be used as drug targets?
The last answer for the answer is, of course, any kind of virulence factor that is required for parasite survival. If you interfere with the expression of this, of course, it's absolutely very interesting drug target. Now, we cannot add factors or apply the environment in culture that is applied in the macrophage, because we just bluntly don't know enough about the biology. One thing that is clear is that the parasites is so challenged in the macrophage due to the toxicity of the macrophage. We need at least to reproduce some of that toxicity. Now, one easy way to do this is using minimal medium, because we realized in various proteomic screens comparing one and the other that actually the xenics lose certain enzymes required for de novo amino acid synthesis, because we feed them with amino acids. So we have to stop doing that. Intuitively, what we want to do is to attain the best parasite growth and the highest density. That is wrong, because that's how you select out the fastest growing that underwent all kinds of genetic modification. We need the ones that just grow and are challenged constantly not to evolve too fast.
Okay. Another question?
Thank you, Gerald, for this fantastic talk. I want to ask you something. What is your opinion about epigenetics in Leishmania? The role of histones, let's say, and H1.
How much time do I have? Few minutes, okay. This is a question that we're actually right now working actively on, because you've seen that we sequence genomes. You've seen we sequence transcriptomes. And comparing the genomes with the transcriptomes, we realize a very interesting phenomenon. Just because you have 3 copies of a chromosome does not mean that you have increased abundance of the respective transcripts all along this chromosome. And this we've seen with all the aneuploidies. There is a lack of correlation between amplifying a chromosome and having more transcripts. Now, how is this working? One way would be, of course, to think that RNAs are degraded because there's too much RNA. Another way to think is that if there is too many chromosomes, in other organisms, there is a mechanism called dosage compensation. Like the X chromosome inactivation is a classical case of dosage compensation. And so maybe epigenetics in Leishmania is a tool for dosage compensation. So what happens is anytime there's an amplification due to drug resistance phenotype, not everything on this chromosome is beneficial. So the parasite can select what do I keep up and what do I actually lock down epigenetically.
Thank you very much.
We still have time for a few other questions. No questions? Good. Right. Thank you, Gerald.